I will today talk about uh, attempts of combination of dark fermentation, biophotolysis, and microbial electrolysis cell for enhancing hydrogen production. The work was published in Biomass and Bioenergy in 2023, in recent published in 18th of June. And this work is written by Vietnam and Korean cooperation by Professor Tuan Long Nguyen from Dui Tan University and by Professor Phan Kang Ting Nguyen from Gashon University in Korea. The work is using first hyacinthus waste from wastewater and the by microalgae from Vietnam, which is these microalgae which are for seaside of coastline of Vietnam natural occurring. So we don't have dangerous risk of introducing new plants. It's a macroalgae which are currently living in Vietnam. So that is a good thing, which is firstly the wastewater is using from free bacteria from free macroalgae, and this wastewater from hyacinth is from so much with wastewater and communist base, and this is was firstly by three types of algae from Sargassum, Uva, and Gracilia, and from this the highest production was by Sargassum. Then the waste, the leaves and water untreated, is then going to dark fermentation, and then from dark fermentation the rest, which is undigested, is broken by microbial electrolysis cell. So firstly, what is macroalgae? Macroalgae are big, very multi-class, multi-cellular plants which spill water to hydrogen. Using wastewater, they can absorb also during this stage some hard or heavy metals, so they can be as biosolvent also. The rest is an undigested part of wastewater is then split to reactor 300 milliliters firstly. And this is used this as chloroform was used as stress agent for dark fermentation bacteria and also for preparing microbial electrolysis cell, because both of them needs to be sequenced, because these pseudomonas occurring also from wastewater, and for wastewater was taken also inoculum. So these are two types, because before was also used dark fermentation with heat treated and also some with chloroform to see how chloroform addition works. As a result, the sargassum polycystin was the highest production of hydrogen. And then the gas sealed was even 400 milliliters per gram of waste. The energy was also the highest. Also, after this was 3 volt and for match, and hydrogen for microbial cell started when reached a microbial cell 0.14 voltage. So these are the waste material that can be split into hydrogen. The chloride concentration was from 0 to 50 ppm, and the gas yield was highest if this chloroform was in ppm. Only the disadvantage of using chloroform increased hydrogen production but decreased acetic acid formation. Most rest was butyric acid. So for optimization of dark fermentation wasn't a very good idea, but a good thing, care per treatment. But for production of hydrogen by microbial cell was very good because it increased, because easily microbial cell can split butyric acid. So this can be optimized. And result was that from all waste, from gram of waste was 700 milliliters of hydrogen. So with 20 milliliters error. So quite big error, but it seems to be uh, mm, uh, 5%, lower than 5%, so still very enough. And this shows that can be combined, and this is successful for high production. Here is used as Vietnam example, which use only native plants, and this shall cause no trouble when comes to the natural world because they have natural herbivorous animals that will make. So this is the example how we could develop. We should seek for plants and biofilters that were in inhabitants of local, and this helps to control. And this shows that it's very good idea that can be combined free many hydrogen production. And this algae waste can be also used as polymers, which was not mentioned because only work is of optimizing hydrogen, not waste. So this list for sure can be used of algae for polymer and waste, and we over useful as Aztec taxi because this algae can be also so this can be as water wastewater cleaning and very high optimizing. So I think very good idea was is here published by Professor by Korean and Vietnam from Professor Nguyen. Thank you very much for giving me a voice.